DNA is like the instruction manual for building and running every part of our body. But it can get damaged by things like sunlight, harmful chemicals, or even natural processes inside our cells. This damage is like tears, stains, or missing words in a book. To keep the manual usable, our body has repair systems that work like editors and fixers. For small errors like a typo in a word, a tool called Excision Repair, or BR, removes the mistake and replaces it with the correct letter. For bigger problems, like a smudged or damaged paragraph caused by UV rays, the body uses Nucleotide Excision Repair, or NER, to cut out the damaged section and replace it using an undamaged part of the manual as a guide. If there's a major tear, where both sides of the DNA are broken, the body can use homologous recombination, a precise process that copies a section from another backup page to fix the damage perfectly. Alternatively, it might use non-homologous end joining, which quickly tapes the broken ends together. This works in a pinch, but might leave a few words out or add extra ones. For some specific types of damage, like a wrinkle in the paper, special tools like direct repair enzymes smooth it out without needing to cut or replace anything. These repair systems work constantly to keep the DNA manual accurate and functional, preventing problems like cancer, aging, and diseases caused by damaged instructions. Without these repair mechanisms, our cells would break down, and the body wouldn't be able to function properly.
DNA serves as the blueprint for life, but it is constantly exposed to damage from environmental factors like UV radiation, harmful chemicals, and internal cellular processes. These damages can result in mutations, cellular dysfunction, or diseases such as cancer. To combat this, the body has evolved sophisticated repair mechanisms to identify and correct damage at the molecular level. These repair systems are critical for maintaining genomic stability and ensuring normal cellular function. Exposure to UV radiation can cause DNA damage in skin cells, leading to mutations. Repair mechanisms within the body actively address and fix this damage, preventing potentially harmful consequences like skin cancer. DNA repair is like proofreading a manuscript, ensuring all errors are caught and corrected to maintain the original meaning. DNA damage can take various forms, including single-strand breaks, SSBs, double-strand breaks, DSBs, base damage, and crosslinks. Damage can arise from external sources like radiation or chemicals, and internal processes such as oxidative stress from metabolism. Cigarette smoke contains toxins that cause base damage in DNA, potentially leading to mutations if not repaired. DNA damage is like tears, smudges, or stains on a delicate fabric, requiring careful mending to restore its integrity. Base excision repair, BER, is a precise repair mechanism for small-scale damage, such as single-base changes caused by oxidation or alkylation. Specialized enzymes called DNA glycosylases detect and remove damaged bases, leaving an empty site. The gap is then filled with the correct nucleotide by DNA polymerase and sealed by DNA ligase, restoring the DNA's integrity. Reactive oxygen species generated during metabolism can damage DNA bases. BER repairs these lesions, preventing potential mutations. BER is like using an eraser to correct a small typo in a written document before rewriting it correctly. Nucleotide excision repair addresses bulky, helix-distorting lesions in DNA, such as those caused by UV-induced thymine dimers. This mechanism excises a small strand of DNA containing the damage and uses the undamaged strand as a template to synthesize a new, accurate segment. A failure in NER can lead to diseases such as xeroderma pigmentosum, where individuals are highly sensitive to sunlight due to their inability to repair UV-induced damage. Example: NER repairs DNA damage caused by UV radiation, preventing sunburn from leading to dangerous mutations. Analogy: NER is like cutting out a damaged section of fabric and sewing in a matching patch. Mismatch repair, MMR, corrects errors made during DNA replication, such as mismatched base pairs. Proteins detect the mismatch, remove the erroneous section, and replace it with the correct sequence using the undamaged strand as a template. This process ensures high fidelity during cell division. Defects in MMR genes are associated with certain cancers, such as Lynch syndrome. Example: if adenine pairs with cytosine instead of thymine during replication, MMR corrects the mistake to maintain genetic accuracy. Analogy: MMR is like a spell checker, catching typos during writing to ensure the final document is flawless. Homologous recombination, HR, repairs double-strand breaks, DSBs, using an identical or nearly identical DNA sequence as a template. This error-free repair mechanism restores the original DNA sequence and typically occurs during the S and G2 phases of the cell cycle when a sister chromatid is available. HR is essential for genome stability and plays a key role in preventing cancer by accurately repairing severe DNA damage. Example: HR fixes breaks caused by ionizing radiation, such as X-rays, preserving the integrity of the DNA. Analogy: HR is like photocopying an intact page from a backup book to replace a torn page in the original. Non-homologous end joining, NHEJ, is a repair mechanism that directly joins broken DNA ends without using a template. It is a faster but less accurate process compared to HR, as it can result in small insertions or deletions, potentially causing mutations. NHEJ is particularly active in non-dividing cells. For example, NHEJ repairs DNA breaks caused by oxidative stress or mechanical forces in cells that are not actively replicating. Certain types of DNA damage can be directly reversed without cutting or replacing DNA. For example, photolyase enzymes repair UV-induced thymine dimers by using light energy to break the abnormal bonds. Humans lack photolyase but use other enzymes, such as alkyl transferases, to remove damaging chemical groups from DNA. Direct repair is highly specific and efficient, targeting particular forms of damage without the need for complex pathways. Photolyase in bacteria repairs UV-induced damage, enabling survival in sunlight. Direct repair is like ironing out a wrinkle in fabric without needing to cut or replace any material. DNA repair is vital for maintaining genetic stability and preventing diseases such as cancer, premature aging, and genetic disorders. Inefficiencies or failures in DNA repair mechanisms lead to the accumulation of mutations, disrupting cellular functions. Studying these mechanisms has enabled advances in cancer therapy, including the development of drugs that target specific repair pathways, such as PARP inhibitors. DNA repair also plays a role in aging, as repair efficiency declines over time, contributing to age-related diseases. PARP inhibitors are used to exploit the vulnerabilities of cancer cells that rely heavily on error-prone repair mechanisms like NHEJ. DNA repair is like regular maintenance on a machine. Without it, wear and tear accumulate,